Okay, to finish up, before we do 9.5 fermentation and anaerobic respiration, I left off with talking about brown fat, which is interesting. It's a type of material. It has lots and lots of mitochondria. And what it does, it has a channel protein called an uncoupling protein that allows the flow of hydrogens down the concentration gradient, just like it would before. But instead of it generating AT, it doesn't generate ATP, and it just kind of flows through easily, but yet it still generates heat because that's a waste product, and it keeps the body of the organism at a level that it is sustainable. So, for example, this would be in hibernating mammals. Even though the body temperature is lower and metabolism is slowed down, you still can't freeze. What else is really important, or I thought was kind of funny when I was thinking about this brown fat, would be like the fat bodies that we saw in frogs. Those aren't necessarily brown fat, but having that fat helps. Maybe it does the same thing as it does with brown fat, where it keeps the body temperature, even though frogs are obviously they're cold-blooded and not warm-blooded, but you still have to maintain body temperature. And they talk about newborns, which makes a lot of sense. You get those little chubby, the chubby cheeks and stuff. Babies have brown fat to help maintain body heat, which I thought was interesting. That makes a little bit more sense when you think about it. Babies don't hibernate, yet they sure act like they do. They sleep a lot. They do grow, so the metabolism part, but they have to be able to come out, and they're able to sustain and have very little food because their stomachs are so tiny that they're able to maintain equilibrium even with just a very small amount of food that they're actually given through, whether it be formula or breast milk or whatever. Okay, fermentation, anaerobic respiration, still producing ATP without the use of oxygen. Remember, oxygen is part of the electron transport chain. If we don't have it, the electron transport chain ceases to operate. Glycolysis can couple with fermentation or anaerobic respiration to produce ATP because we're still doing glycolysis no matter what. The anaerobic respiration actually uses a different electron acceptor other than oxygen, for example. And I wanted to see if I could show you. Let me switch programs here real quick. I wanted to see, I wanted to show you if this is where I saw this. Is it here? Maybe it wasn't at this website. There was a website. I'm going to have to find it again. There's a website that actually showed a place. I'll come back to this website. This is showing some different molecules. We'll talk about this in class. But I was looking at, they gave me an examples of different organisms and the different electron acceptors at the end, which was kind of interesting. Besides oxygen, sulfate would be a good one that accepts it at the end. Okay, and fermentation uses substrate level phosphorylation, which we know, again, transferring a phosphate to a substrate and then generating ATP. And with both of these, we know that with substrate level phosphorylation, we don't get a lot of ATP generated from that. In fermentation, so we have two types of fermentation, consists of glycolysis plus the reactions that actually regenerate NAD+. And this is why, in order to continue to do glycolysis, NAD+ has to be regenerated in order to start the process. There are some great pictures on right here. These are the pictures that I wanted to show you, as glycolysis will keep going as long as you have more NADs to actually make more NADHs. And in alcohol fermentation, you get ethanol in 2 steps, and of course the release of carbon dioxide. And alcohol fermentation, you could say, well, this is yeast, brewing beer, winemaking, baking, all of those things, releasing carbon dioxide. And that would be, so we make 2 acetaldehydes, which makes 2 ethanols. Here's an example. So we're going to stay right. We're going to stay right about here. Let me see if I can play this.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Okay. Forward. This is the part where there we go. So this is showing you, here it is, alcohol fermentation. So the acetaldehydes, again, 2-pyruvate converted to 2 acetaldehydes. We're going to release carbon dioxide here and then make 2 ethanols. And we'll go back to lactate in a second. Lactate for lactic acid fermentation, pyruvate's reduced to NADH, forming lactate, but you don't get a release of carbon dioxide. And this can be used by fungi to make cheese and yogurt. And then, of course, the classic is with our muscle cells when O2 is scarce. And when you actually go into something called oxygen debt, and an oxygen debt, that is the amount of oxygen you have to pay back to the muscles to go from ADP back to ATP. There's also things called creatine phosphate that you have to regenerate. And you have to regenerate things in the liver. So it's a whole big cycle of things that you have to repay to everybody after. And the lactic acid will cause soreness, but it'll eventually go away. Notice no production of carbon dioxide here. And comparing fermentation with anaerobic and aerobic respiration during glycolysis, you have 2 ATP. In all 3, of course, fermentation, anaerobic, aerobic respiration, NAD is your main oxidizing agent. That's the thing that gets reduced. Remember, you go NAD plus to NADH. You have electron acceptors, except in the anaerobic respiration, you have something different than oxygen. In an organic molecule, in fermentation, you might just stick with glycolysis. Cellular respiration produces about 32 total. In fermentation, you only get 2. So the big question is, there are organisms out there that cannot survive in oxygen environments. Those are called obligate anaerobes. So anaerobe means without oxygen. So this is obligate, kind of sounds like something you have to do. They cannot survive in the presence of oxygen. With that being said, they will rely on glycolysis completely, and that's all they're going to do. Yeast and some bacteria are facultative anaerobes, meaning they can survive either using fermentation or cellular. If for some reason the environment doesn't allow oxygen, well, then you can flip back and do the other way. So this is really an adaptation, I think, from this, giving rise to this, that you can go either way. So you have 2 different alternatives. So with the pyruvate, you can either go on to do the citric acid cycle and electron transport chain, or you can stick with just glycolysis. And here's the example here. Now, when you think about it, we talked about this in class, that glycolysis is used whether there's oxygen or no oxygen. So therefore, it's got to be one of the most ancient processes because, again, it can be done in the presence of oxygen or without the presence of oxygen. So obviously, the rest of the chain was later because of the presence of oxygen. Something else that I want to point out here too. If you have an organism that just solely relies on glycolysis, you're only making 2 ATP, and you have to have 2 ATP to run whatever organism it is, then you better have a whole bunch of glycolysis or glucose coming in and a whole bunch of regenerating of NAD. Otherwise, the process is going to stop. that would be your only area to get ATP, for example, in this case. So 2 at every turn of glucose. So you might have a really super influx of glucose just to be able to survive. And maybe the organisms that just rely on that are more primitive. Maybe they are not, maybe they don't have as many emergent properties as something that is more complex, which I think is interesting. You know, when we're thinking about the big scheme of things. Okay, glycolysis in the citric acid cycle. So we've been talking about glucose, but what about the other pathways? You know, how do fats and how do proteins are digested? Well, you have to think about what's the starting blocks. So it says catabolic pathways, meaning how are you going to break up a cheeseburger, or how are you going to break up that juicy steak in order to get the raw products that have to enter the cycles. For example, proteins have to be broken down to the raw amino acids, and even with that, there has to be some processes that actually strip those amino acids actually of certain chemicals so that we can start the pathway, because lipids, proteins, and of course our sugars can all go through the citric acid cycle. Fats, for example, have to be broken up into glycerol and into fatty acids. The glycerol can later on then become or be transferred and made into glucose and then go through cellular respiration. The fatty acids can actually directly go into acetyl coenzyme A, but they have to be broken down by a process called beta oxidation, which oxidation sounds like a redox reaction. You can actually make fatty acids go to acetyl coenzyme and then go into the citric acid cycle. Interesting too, gram for gram, fats produce way more energy. Unfortunately, that's not the most efficient way. So it might take a little longer. This is why fats aren't necessarily the first ones to be broken down when you really, really need some energy. Something interesting too, beta oxidation produces a byproduct called ketone bodies. So if you fast, for example, you might get fruity smelling breath and urine. That's ketone bodies that's given off. You'll have to ask Mr. Keller about that one. When he went on the Atkins diet the first time, he metabolized so much fat he produced ketone bodies. Let me see if I wanted to say, they really didn't talk about amino acids too much. I do want to mention they have a process, and they did mention it in the book. Proteins broken down to amino acids, again, hydrolysis, but there's another pathway that removes some of these groups off, and it would be called deamination, which sounds like deaminate, like amino. And then a couple of things, those parts can then, one of them can go and act as sugar. One of them can go directly and go into the make acetyl coenzyme A, or go directly into the citric acid cycle. And keep in mind that proteins, carbohydrates, and fats can all be stored as fats and then broken down for later use too. So you can get fat off steak, you can get fat off cheeseburgers, you can get fat off too much pasta. But pretty much we lead to the same pathways at all times. And the body uses some molecules to build. We've been talking about cutting things down. We can also build up things. So small molecules can come directly from food, from glycolysis, or from citric acid cycle, and we can use those smaller molecules to actually make things. So it's not all breaking things down. It can actually make things. Something else to keep in mind, too, as we're doing all of these things, that the body is very efficient. If you already have too much of something, it will slow it down. How does it slow it down? Well, it has those feedback mechanisms, those allosteric regulators that can come back and slow down a portion of this catabolism, meaning breaking glucose down at some point. Just like we talked about ATP, we can slow that down. AMP is a adenosine monophosphate, I believe, can actually come in, and that phosphofructokinase was one of those steps in glycolysis. We can stop that right there. I believe that was either step 3, somewhere around there. I can't remember. Citrate, for example, produced in the citric acid cycle, that was like step number 1, oxaloacetate plus acetyl coenzyme gave you citrate, can actually go back and inhibit this. ATP, too much ATP at this point from the citric acid cycle, though we don't have much, can actually go back and inhibit. So this guy right here is affected by all these allosteric inhibitors, because if we don't need the energy, why are you making more? It just doesn't make sense. So here you go, inputs, and there's your outputs, and I believe this was some of the answers to some of the questions at the end. I'll put up the clicker questions at the end. Make sure to look at your concept checks and check those in the back of the book, and come in with any questions that you may have.